This is Mrs. Palmer-Quay, and this is the second video for Unit 3. In this video, I want to just review some of the principles of genetics, which hopefully you remember from your biology class, and talk about a few of the familiar human genetic disorders. Our modern understanding of genetics was based very much on the work of Gregor Mendel, who was a monk in the 1800s who did a lot of work studying pea plants. He was very interested in trying to determine how things were passed from parents to offspring, what were the rules on how these particular traits or characteristics of the pea plants were inherited, and he came up with several laws of inheritance which helped determine to predict what you would see in an offspring. His work, though, was not recognized by his contemporaries. It wasn't really until the 20th century that we were able to take what he had done and apply it to our greater understanding of biology. So these are some of the words that you need to get a handle on or remember when it comes to talking about genetics. One word, or a very important word, is trait, which is any characteristic that can be passed from parent to offspring. This can be something very obvious, like someone's height or skin or eye color. It also can be whether they can taste a particular chemical in food, or if they have a particular allergy, things that might be hidden from first view. But all of these things are considered traits. And then, of course, the passing of traits from parents to offspring is known as heredity. We now know these are determined by genes, the sequence of DNA that codes for protein that causes a particular trait, or several proteins working together. In Mendel's days, he just called them particles because he wasn't quite sure what was going on. He just knew something was influencing the passing on of these traits. Alleles then become the word that refer to the various forms of a particular gene for a particular trait. So you may have, in Mendel's time, he understood that there were two alleles for any particular trait. One allele was understood to be dominant, it was stronger, it would be expressed whether it was in the pure state or in the hybrid state, and it was shown as a capital letter. And the other one was recessive. It was a gene that was expressed only if it was the only allele present, if there were no dominant alleles. And recessive alleles were shown by a lowercase letter. So then we get a genotype, which was the gene combination that was for this particular trait, whether it be the capital letters or small letters, it would be what was found in the genes. But compared to the genotype, we have a phenotype, which is the physical expression of that genotype. That is what you are seeing in a person that is leading you to believe, or in an organism, what the genotype is. So the genotype is what it actually is in the genes. The phenotype is what is being shown. So genotypes come as homozygous when we're talking, again, the dominant and recessive versions. So this would be a homozygous dominant genotype, but here we have a homozygous recessive genotype. These are also sometimes called pure strains or the pure parents. Then heterozygous genotypes are when it's a hybrid situation, so that you have one allele that's dominant and one allele is recessive. So just to give you an example with Mendel and his peas, one of the things he looked at was pea color. He also looked at height and whether the pea seeds were wrinkled or smooth, or if they were yellow or green. He looked at 7 or 8 traits that he was tracing. And so here he had two parents. This is the homozygous pure parent with the dominant purple flower. Here we have a homozygous parent with the recessive white flower. When they were bred together and the pea seeds from their offspring were collected and planted, they were known as the F1 generation, and they all were purple flowers. And you can see this is the heterozygous genotype here, because one parent is contributing only the dominant allele and the other parent contributes only the recessive allele. And this little square thing is a Punnett square, which is what can be used to calculate out the percentages, the ratios of the different potential offspring. Then if Mendel took these F1 generation peas and bred one of them, the heterozygous pea colored purple, and this other heterozygous pea, put those two together and collected the offspring known as the F2 generation and planted them and saw what colors, we could see that this white flower was reappearing, because when you're combining these two heterozygotes, you have both the dominant and the recessive allele being contributed by both parents. So there's a 25% a chance that one of the offspring will be the homozygous recessive and show up this pea color. So this was the kind of work that Mendel did, and really provided a lot of the groundwork for understanding how things are inherited. But people aren't peas, and things in the human world are a little bit more complicated. This also applies to many other animals. Perhaps God directed Mendel to start with peas because he really picked traits that were either or. They were dominant or recessive. But we have situations where there's codominance or incomplete dominance, multiple alleles. Things get much more complicated. In codominance, both traits are expressed if the alleles are there. So in blood typing in humans, we have type A blood, type B blood, and if the alleles for A and B are inherited, then someone will have type AB blood. It's not as common as the other types. It is a more rare situation, but it does exist because A or B, neither one dominates. They both are there. They're both being expressed in the blood. And there also is a recessive in blood typing. So this has got 3 alleles involved. And so you can have someone who's got the homozygous recessive state would be having type O blood. In incomplete dominance, the traits are blended, and often you get much more than 2 or 3 genes involved. I think there's 16 genes involved in skin color, and they're just really beginning to understand how these things happen. So the color of your eyes and the color of your skin, the color of your hair, the texture of your hair, the height, how tall you are, these are all based on this situation where you're inheriting a number of genes that affect the expression of that particular trait, and the various genes, their expressions are being blended together. That's why it's very hard to predict what offspring will look like. That you're a blend from your parents, and different children are different blended combinations. And the other thing about human organisms and other organisms is that there are many environmental factors that go on to influence how your genotype is expressed. So first of all, the inheritance is not straightforward recessive and dominant in many, many cases. And what you are eating, or how active you are, or if you're exposed to a particular toxin like cigarette smoke, or the level of stress in your life, will all influence gene expression from things as kind of clear to see as how old you look, how wrinkled your face is, to whether you get a particular type of cancer or not. We're finding more and more how important these environmental factors are. So these are known as epigenetic factors, or the epigenome. So you not only have a genome that refers to your genes, but you have an epigenome going on in your life that determines how your genes are being expressed. And the website that I've listed here has a number of interesting videos and interactives if you want to get a little better idea about epigenetics and some of the things that we're learning. So after that quick review, I just want to talk about the few of the perhaps more well-known genetic diseases or genetic disorders, just kind of review some of this stuff. We'll start here with the human karyotype. This is a nice little picture of somebody's chromosomes. And the karyotypes are made right at the point of mitosis, because the only time these chromosomes are going to coil up so nicely like this will be when the cell is getting ready to divide, just before, after prophase and just before metaphase. And so these little X-shaped chromosomes, the chromosomes have already duplicated. If they've just been, if they were just lined up without being part of mitosis, if we'd managed to get the chromatin to coil up, you would just see straight lines, not these X. But here we've got a duplicate of the individual chromosomes held together here in the center by the centromere. So you can see that there are 23 pairs of chromosomes, 46 total chromosomes, but they come together in pairs. And on any particular chromosome, so say on this spot on chromosome 1, will be the same allele will be found on the same part of the other chromosome 1. So this particular chromosome perhaps came from the father, and this one came from the mother. So this allele right here is the father's contribution, and this is the mother's allele. And so under normal circumstances, both of these chromosomes, whether it be chromosome 2 or chromosome 7, they will contain alleles for the same traits, so that you're getting a duplicate set of alleles for any particular trait, one from your mother and one from your father. There's also a distinction between the first 22 chromosome pairs and this last one. Chromosome pair 23 is known as the sex chromosome, and this is what determines whether you're male or female. If you're female, you receive 2 X's, but you can see from here we've got just an X, 1 X, and then this little Y chromosome. So this karyotype came from a man. And then these 22 chromosomes are known as the autosomes, everything else but the sex chromosome. And so some of your genetic disorders are coming because of genes that are on one of these autosomal chromosomes, one of the first 22, and sometimes it's a chromosome that is the sex chromosome. This chart shows some of the autosomal disorders, something from those first 22 chromosomes, and we have identified in many cases what particular chromosome these disorders are coming from because of the Human Genome Project, and we've mapped out the entire human genome. We've been able to identify many connections between disorders and genes. So this first list here, these are all disorders that are caused by recessive alleles. So someone would have received 2 copies of the recessive allele, one from each parent. The parents may have been heterozygous, so they wouldn't be known as a carrier. They're not going to be showing this recessive allele because they have this dominant one that will override it. But the offspring then will inherit both recessive alleles. And so situations like cystic fibrosis, albino, Tay-Sachs are all caused by recessive alleles. PKU is not a disease we hear about very often because we're testing babies in the hospital for that. It can be managed by dietary changes, and so it's not the problem that it used to be. And then there are a few disorders listed here that come from dominant alleles, so someone who is either homozygous or heterozygous will show this particular disorder. In some of these disorders, such as the dwarfism that's mentioned here, achondroplasia, this is only the heterozygous form because the homozygous form is lethal before birth. So that is why parents, if both parents have dwarfism, they can have children of normal height and then children of the smaller height because they are all, the only possible way to be alive is to be heterozygous. Homozygous do not survive birth. They don't survive gestation. Another, Huntington's disease, this particular disease doesn't manifest itself until someone is in middle age. And so this is why it is staying in the population, because people are heterozygous or homozygous for it, is a dominant allele, but you don't know it until you possibly already have had children. We now have a test to have people can find out whether they're carrying this particular disease when they are young and to decide whether they want to have children or not. And then we've got one example of a codominant allele, and that is sickle cell disease. So you know, in sickle cell anemia, you see in the blood slide there are normally shaped blood cells and then there are sickled blood cells. So this is a hint that it's a codominant situation because both are being expressed. If it were only sickled cells, probably again you'd have a situation where the human could not survive. Then a special case of the genetic disorders are ones that are carried on the X chromosome, sex-linked diseases. In this case, the mother is often a carrier and is only, having one bad gene is being covered over by the fact that she's got another X chromosome with a good allele, and the male, though, only receives one X chromosome, and so he will be affected. So we just see this as a pictorial pedigree showing crossing an affected or unaffected father with a carrier mother. You will see the son being affected because he's receiving the X chromosome from his mother and he has no other protective chromosome. But his sister might be carrying the disease, but she will not show it. It will come out in her children like it did with her mother. One not very horrible sex-linked disorder is color blindness. the red-green color blindness is on that particular allele is on the X chromosome, and so boys that are colorblind are receiving that X chromosome from their mother. And so I found this nice illustration that shows you the difference between what someone would see with predominantly red and green colors under normal vision and having colorblind vision. Of course, people who are red-green colorblind are not able to look at the traffic lights and see the colors. So you test yourself. Do you know what color is on top and which color is on bottom? Which one is red and which one is green? They have to memorize it because the color doesn't show up. A very, very well-known sex-linked disease is hemophilia. Hemophilia, that is the bleeding disease, and this is a pedigree of the royal family from Queen Victoria down several generations. She had a spontaneous mutation. Hemophilia was not present in her family prior to her, but she ended up being a carrier female, and you can see she passed it on to 2 of her daughters and 1 son that suffered from hemophilia. Her daughters went on to pass it on to their sons and their daughters down to the Tsar in Russia, because Queen Victoria's grandchildren, kind of spread throughout Europe, and in Prussia, and then also the royal family in Spain. And I believe there's some possibility that hemophilia from Queen Victoria is still present in some of the relatives of the royal family in Spain, but in the current present British royal family, there is not any hemophilia anymore. So this is another very familiar sex-linked disease. The last type of genetic disorder I want to briefly discuss are non-disjunction disorders. These are disorders that arise because chromosomes do not separate the way they need to when gametes or sex cells are being produced. If you remember back from biology, this is the process of meiosis, and there are 2 levels of separation that have to occur so that the sex cells end up with half the amount of chromosomes, so that you can have one from the male, one from the mother, one from the father, come together to form the fertilized zygote that will then develop into the embryo. So when things do not separate the way they are supposed to, you can have sex cells that have 1 too many chromosomes, and if there's 1 too many on this side, also at the same time you're producing ones that have 1 too few. But there are certain conditions that the embryo that has been fertilized with one of these sex cells can develop and can be relatively viable. Probably the most familiar non-disjunction disorder is Down syndrome, which comes from an extra chromosome 21, one of the autosomal chromosomes. And you probably have seen or know people, perhaps somebody personally who has Down syndrome. They can go on to lead productive lives and be part of the community. The only type of non-disjunction disorder that I am aware of that is viable when only 1 chromosome is inherited is one that is called Turner syndrome, when a woman only inherits 1 X chromosome instead of 2. She, of course, in any of these non-disjunction disorders, and there are some other ones as well, there will be repercussions, there will be problems with body systems. Often there's developmental delays and mental retardation, but in many cases the person can go on to be part of society and play a role in their family. So this finishes the brief overview of genetics. Hopefully this reminds you of the stuff that you learned back in biology. We're just going to be running into these ideas about genetics and inheritance, especially as we're looking at individual organ systems and how some disorders within those organ systems are affected by genes or mutations or other situations that are closely connected to heredity. There's one more video coming for this unit that will cover tissues and look at some of the other topics that are specific to the recommended learning targets for honor students.